Okay guys, what I'd like to do is get started. Since we're now already just a couple of minutes late, I'm not too worried about that, since as you probably have realised, I get to give you 2 in a row, which has just got to be not good fun for all of us, but anyway. So what I'm going to talk to you about now is what goes on in a cell and what goes on in a cell as it's growing and dividing normally, and then what is it that goes awry, what are the actual molecular underpinnings that lead to cancer. So that's what the theme of the lecture is going to be: normal growth, which is responsive in that cells grow when they should and stop growing when they should. And of course that's always a desirable output. I feel it's most unfortunate that adipose tissue doesn't always know when to stop growing, but apparently they say that's my fault, not its, but I don't believe that. And then what does go wrong to lead to the malignant phenotype. So the first thing I want to remind you about is this is what it looks like when things are going normally.
As this capsule, taken from a lily, prepares for division, the chromosomes first come down. Next, the mitotic spindle lines them up in the center of the cell.
But you must admit, they are quite sinuous, the chromosomes, as they're moving, right?
Anapase transition, the sister chromatids of every chromosome pair separate suddenly in striking synchrony. The chromosomes are pulled along the microtubules of the spindle to opposite ends of the cell. After chromosome separation, membrane vesicles line up in the center and fuse with each other to form the neoplasm membrane that separates the 2 daughter cells. At telophase, the chromosomes decondense in the newly formed nuclei.
Okay, does anyone recognise the music? Ravel's Bolero, very good, very, I think, quite sensuous music. So, that's just to remind you of what goes on in a normal cell as it goes through cell division. As we look at mammalian cell division, what we find are the cells actually go through a very highly regulated and highly organized process. So what scientists have worked out is that bit that you just looked at there is actually the smallest part of what goes on in the life of a cell. If we actually look at the whole process, we see that many things in life, it's cyclical, and so you actually have this cycle which is divided up into these different phases. And so we have a stage here called G1. This is the first growth phase, where the cells are actively growing and they're metabolizing. If they receive the appropriate signals, called growth factors, and they must receive these external signals, they will then move from this growth phase, the first growth phase of G1, into the S phase. This is where they actually replicate their DNA. They don't replicate their DNA in mitosis, they separate the chromosomes there. So in this growth phase here, they've obviously had to accumulate all the raw materials they need to replicate the DNA. If you have 6 billion bases, nucleotides, comprising the DNA of a human cell and you want to replicate them, you've got to bring in another 6 billion nucleotides and assemble them. It's not a trivial process when you think about the organizational skills. If you wanted to copy 6 billion nucleotides and you wanted to get it right, and that's what's happening in this phase of the cell cycle. It's a huge process. So the cell waits till it gets this growth, these external signals. It allows them to go past what we call the restriction point. Cells won't move through the cell cycle past here till there are adequate nutrient supplies available and this external growth signal that says go. They then go and then they start the onerous process of replicating all their DNA. They need to decondense it, replicate it all. The cell now is getting, obviously starting to look bigger, goes into a second growth phase because you not only have to replicate the DNA, which is what we tend to think about when we think about cells dividing, because we can see that happening, the actual chromosomes dividing up here, we can see that visibly. But all the other organelles have to be replicated too. When the cell divides, it needs new mitochondria, needs new membranes, new endoplasmic reticulum, new ribosomes. So all of that has to be started to be recruited too. And that's happening here in this second growth phase. Then when all of that's assembled, the cell goes into the interesting part, the part we can see under the microscope called mitosis that you know about. And then you actually get cell division. And then the cell can go back, as it grows, it's now smaller, can now start growing, and it can go back through that whole cycle again if there are still adequate growth factors. Cells can also do what we call exit the growth phase. And that is, it can actually come out of here and actually go into what we call the G0 phase of the cell cycle. And that is something that it can also do. And that's where cells don't continue to divide anymore. And you know there are many cells in our body that don't continue to divide, and so they actually don't continue to go through this cyclic process, but they do continue to metabolize and are active. And many cells in the adult obviously are in this stage. They're not actively dividing, but they are still actively metabolizing and carrying out functions. And we say they're in the G0 phase of the cell cycle. If there is an appropriate stimulus, so for instance, if you cut your hand, you're then going to stimulate, you're going to get the appropriate growth factors will come along and actually stimulate the dermal stem cells to start dividing, to start healing and replacing the damaged skin, then grows, and then it can go back into G0. So that's what we call it. Now this whole process we call the cell cycle. And in normal adult human cells, if they're stimulated to divide, it takes on average about 24 hours to go through that whole cycle. So I showed you here. Now what intrigues scientists is, what is it that makes a cell move through their cell cycle? What is it that makes it actually do that, and that makes it do it in the right order? I mean, it makes sense. I lay it out as a nice, neat diagram here, and you think, oh, that's logical. First we grow, then we replicate stuff, we grow a bit more, and then we divide. Why does it happen in that order? Why don't they get this external signal and then go straight into mitosis? So scientists were quite intrigued by this, and to try and work it out, they did a very elegant experiment. And what they did was they took two different cell types, a human one and muntjac cells, and what they did is they fused them together. And you can do this. You can add a chemical that will cause the cell membranes to actually fuse. And they took a cell that was already in mitosis, and they fused it with a cell that was in the G1 phase of the cell cycle. So when they fused and merged the two membranes, all the cytoplasm within those cells could actually mix and merge. Now, the G1 cell still has a nuclear membrane, but remember, when you go into mitosis, the nuclear membrane has to break down so that we can separate the chromosomes. So no nuclear membrane in the mitotic cells. And what they found when they fused those two together was the cell that was in mitosis continued on its normal progress through mitosis, but the cell that was in G1, it induced the nucleus to break down and the chromosomes to start condensing, as if they were getting ready to go into mitosis, even though they had not replicated their DNA. Excuse me. So you can see here we have the metaphase, the cells, the chromosomes from the mitotic cell, and here you can see this G1 cell, the chromosomes are now starting to condense. Instead of looking like this, where they're decondensed, they start to condense. This implies there is some diffusible factor, some factor that's present in the mitotic cell that can induce mitosis, even in a cell that's still in the G1 phase, hasn't replicated its DNA. It starts to act as though it's going into mitosis. They repeated the experiment, and they did it with a cell that was in mitotic cell and a cell that's in S phase, where it's replicating its DNA. And they did the same experiment, and again, the mitotic cell induced the premature condensation of the chromosomes. That is, it made the chromosomes start to condense forced to start, the cell is moving into the mitotic phase. And you can see here they're all look like they're all kind of pulverized, the ones from the S phase cell, because the S phase cell is in the middle of replicating its DNA. And remember, we replicate DNA in short bits, so you can see them all decondensed. And then finally what we have is they took a mitotic cell and they fused it with cells that were in the G2 phase of the cell cycle, and again you got this induced condensation of the chromosomes. So what this inferred was there was some factor present here in the mitotic cells that would start to induce mitosis in any cell that it was fused with, no matter what stage of the cell cycle it was in. And they assumed that therefore there must be some, and it's probably a protein. And so what they did is they started looking to see if they could find proteins whose levels fluctuated with the cell cycle, which made good sense. And indeed what they found was, we're looking, here's the cell going through interphase, that is G1, SG2, all the stages that are not mitosis in the cell cycle, and then the cell goes into mitosis, back into G1, G2, G1, SG2, interphase, the bit between mitosis. Scientists used to think this was the boring bit, nothing's happening here. And you know now, it's G1, they're replicating, then they're going to into S phase, replicate the DNA into G2, but back then that's not what they could see. So, and then back into mitosis. And then they found this factor, they called MPF, mitosis promoting factor, whose levels did indeed peak when the cell was in mitosis, fall when the cell was in interphase, rise, fall, rise, fall. They found another protein called cyclin, whose levels cycled. And what was more interesting about it is you can see this one peaks in the middle of mitosis and then falls as the cell exits mitosis, but this one's, its levels start to rise as the cell goes into mitosis and then falls when it comes back out. So if it's starting to rise, you might think, well, this is the one that's inducing the cell to go into mitosis, and then it falls, but this is the one who's peaking during mitosis. And when they investigated further, I'm summarizing very large amounts of work here, they found in fact that MPF, the mitotic promoting factor, this one here, is actually composed of two proteins. It's composed of that cyclin. So this compound here is actually a component of this. And as its levels rise, as this protein's levels rise, what it then does, it actually associates with another protein that's called a cyclin-dependent kinase. So the cyclin, obviously as its name suggests, it's a protein whose levels cycle with the cell cycle, and it associates with and thereby activates another protein called a cyclin-dependent kinase. Anyone remember what kinases do? They phosphorylate, and you're going to find this is a key theme. We can very markedly alter the activity of a protein if we phosphorylate or dephosphorylate it. So the protein that carries out that activity, the kinase, can have significant impact on what's going on. So the fact that it's cyclin-dependent kinase obviously means that it only can phosphorylate its substrates, it's only active when it's actually associated with the cyclin. If the cyclin's not there, it's not active. When the cyclin levels come up and bind to the cyclin-dependent kinase, that kinase is then activated, and then it starts phosphorylating its key substrates, which are proteins. So they had a look at this and followed it through, and sure enough, that's exactly what's going on. If you actually follow it, it's the fluctuating levels of MPF are what drives the cell into and out of mitosis in the cell cycle. And so you see the cyclin levels start to come up as you get more and more cyclin. They bind to the cyclin-dependent kinase to form the active MPF. It's then active. Its levels must drop so that the cell moves out of mitosis and back into G1 once it's divided. And then as the cell goes through the cell cycle again, the cyclin levels start to come up in G2, associate with the cyclin-dependent kinase to form the active MPF. The cell goes into mitosis, it drops again. And we've shown it here diagrammatically. Here we have the cyclin-dependent kinase is present all the time. Throughout the cell cycle, you have the cyclin-dependent kinase there. In G1 and S, and as you come out through G2, it's not associated with the cyclin, so it's not active. But as the cell moves out of S into G2 and then through into mitosis, the levels of cyclin start to accumulate. They associate with the CDK to form the active MPF, and the cell goes into mitosis. You also need the cell to exit mitosis, so you then need to break down the cyclin. It's the cyclin that gets degraded, its levels drop, plummet, that inactivates the MPF, and now we just have the CDK released. Does that make sense? So this is when they did all these studies, they worked out this is what is pushing the cell through the cell cycle and into mitosis. The fluctuating levels of MPF, driven by the cyclic rise and fall of the cyclin to bind to the CDK to make the active MPF. So, what does MPF do? Well, what it does is it specifically phosphorylates different substrate proteins that are responsible for the mitotic process. So what you have is, once this binds, it changes the conformational shape of the CDK so that it then can phosphorylate its key substrate targets. And some of those key substrate targets that it phosphorylates are the proteins that make up the nuclear membrane. And when they get phosphorylated and thereby acquire a net negative charge on each individual protein that forms the membrane, they repel each other and come apart, and so the nuclear membrane breaks down. They carry out activities like that. They phosphorylate some other activities that help get the microtubule set up so that they can bind to the chromosomes and pull them apart during the mitotic process. So they're the substrates for this MPF that you need to have activated so the cell can go through mitosis. Well, that's fine and dandy. We've got the cell into and out of mitosis by actually activating the cyclin-dependent kinase by forming the active MPF. But guess what? We actually want to get the cell not to just go through mitosis. We actually need it to actually go into the cell cycle as well. So it turns out there are other cyclin-dependent kinases that have their own specific cyclins that bind to them. So the one we've just looked at is this one here, the mitosis, the M-phase promoting factor, and we've just talked about it. But there are other cyclin-dependent kinases that bind to their own specific cyclins to be activated, and then they phosphorylate different substrates that can push the cell through the different phases of the cell cycle. It's the same principle, but different specific activities. So at the beginning of the cell cycle, what you need is this particular CDK to be activated to phosphorylate the proteins you need to move the cell out of the G1 phase and into the S phase of the cell cycle so that you start replicating the DNA. And that's the activity that that particular one carries out. If we actually look at what goes on in mammalian cells, you'll see that it's actually relatively complex. There are different cyclins with different CDK partners. In each case, though, throughout this cell cycle, the principle is the same. You get increasing levels of cyclin D, you get an external growth factor that stimulates the production of cyclin D. The rise in cyclin D means it associates with its partner CDKs to activate them. They also help regulate the production of cyclin E so that it can partner with its partner CDK to move the cell out of the G1 phase of the cell cycle around into the S phase. They also help stimulate the production of cyclin A so that it can partner with its partner CDK so that the cell goes through the S phase and replicates its DNA. Their levels fall at the end of each of those, but one of the activities of this cyclin A CDK2 partnership is to upregulate the production of more cyclin A and cyclin B to partner with CDK1 to then start pushing the cell through into mitosis, and then those levels have to fall. I don't expect you to remember the order and the sequence of each of the cyclins and the different CDKs. Although I'm really tempted to tell you you have to, just because I'm in that kind of a mood. But there's no point for you to have to remember, oh no, I think it was cyclin A was a mitotic no. I'd like you to understand the concept, partly because it's a beautiful piece of organizational skills to fine-tune and regulate this so precisely, but also because it's these going wrong is partly what can lead to cancer. So we have this highly regulated process so that we have it all orchestrated with the rise of the appropriate and different cyclin to then move the cell through each of these stages till we get to cell division. How does this whole thing work? Well, basically what we have, if we have a very nice square cell here, not commonly seen your square cell, but in this diagram we have a square cell. What we have are proteins that span the cell membrane, and those proteins are called receptors. They are usually dimers, you need two of them, and they're not actually hooked together. But when you have a signaling molecule, a growth factor come in, it will interact with one of those receptors and cause it to now dimerize. That activates it, and once it's activated, it's a kinase usually, it will phosphorylate another protein to activate it. It will phosphorylate another protein to activate it on through the cytoplasm until they finally, one of these phosphorylated proteins moves into the nucleus to act as to activate a transcription factor. They're the proteins that bind at the promoter regions of the DNA to turn on the production of proteins, and the proteins that can be turned on are those cell cycle control proteins, the cyclins. When they're appropriately turned on, they then trigger the cell to enter into the cell cycle, and they themselves then trigger the production of the subsequent cyclins that are needed for the cell to go through the cell cycle. Growth factor interacts with the receptor to activate it. Usually as a kinase, it's usually a kinase once it dimerizes together to activate another protein by phosphorylating it. It's usually a kinase that phosphorylates something else. And there's often many proteins in this pathway, so it finally moves through into the nucleus to upregulate a transcription factor, activate that protein to bind the promoter region of the DNA upstream of the gene you want to turn on, which are usually the cell cycle control proteins, to start the cell going through the cell cycle. Sometimes they activate DNA repair proteins to protect the cell. Sometimes they upregulate apoptotic proteins. But what we're focusing on here at the moment is the production of these to then get the cell going through the cell cycle appropriately. There are many steps, and I said they're exquisitely regulated. What I love about nature is, of course, nature also puts checkpoints in just in case something goes wrong. And this is what's so reassuring. First of all, I just want you to see if you can see, I actually downloaded this image from the web. Warning, when you download images from the web. Can anyone see if I point around here where there's something wrong on this diagram that I'd like you to correct? Thank you. See, you have learned something this morning already. Excellent. Even if it's a seeing first gap having G2 on it going, exactly. Please make that G1. That was an easy error to pick up. Just do note when you download things off the web. But I wanted you to have this diagram. I liked it. The cell has 3 cell cycle checkpoints. That is where it will stop, and the whole process won't continue until it's reviewed and everything is, you're absolutely sure everything is okay. The first one is the G1 checkpoint here. The cell size has to be adequate. You have to have sufficient nutrients so that you can replicate the DNA and all the other organelles, and you must have the growth factors present that say it's appropriate. Cells are not of their own volition allowed to sit there and go, you know, slow day, bit boring, nothing on telly. Oh, I know, I'll reproduce. That's what I'll do today. Yep, I'll auto-stimulate and reproduce myself. Great idea. The world needs more of me. That's what I'll do. Put it on Facebook. Tweet. Unless it... as an organism, we need control of individual cells. The organism determines when a cell will replicate by sending the appropriate growth factors to the cell to say, I know you're bored, don't do anything, or I know it's time. We actually need some growth here. Damage to the skin, you can start replicating. Turn off the telly. Get off Facebook, start replicating. So that's what you have, and that's what this one's here for, to block the cell from moving through unless it's appropriate. The cell goes through replicating all its DNA, and you might think there's a checkpoint there, but there isn't. The next checkpoint comes at the end of G2. And this is where you have to check that all the chromosomes have successfully duplicated, all the DNA has been successfully duplicated and replicated, and the organelles. Have we got everything in place before we actually commit to dividing? So DNA is replicated, cells big enough, organelles have all replicated, then the cell moves through to mitosis. But there's still another checkpoint, and here this one's very important. Here, what you want is when the cell divides, you need to be sure that when we have, if this is all, if we've got all the chromosomes all lined up at the nucleus and I'm the little spindles attached to the chromosomes, when they move apart into the two new daughter cells, you really want one copy, you know, of each to go into the daughter cells. You don't want one getting more and one getting less, because that's what really leads to cancer. And so what the cell has is it actually has a wonderful mechanism dependent on those spindle fibers, the microtubules. If there's not equal pressure at the poles, it won't move apart. So if we have this, but one's not attached, the cell won't divide. That's the mitotic checkpoint. It'll stay until this one also attaches, then it goes into division. It's a very important checkpoint. So that's very reassuring for us to know that there are appropriate checkpoints to ensure that the cell actually doesn't divide until it's the right time, until everything is completely ready. Okay, so once the cell's gone through all this process, growth factor, started the production of the appropriate cyclins to stimulate the production of all the other proteins that you need, the appropriate processes, passed the checkpoint, replicated the DNA, got all the other organelles being reproduced, cell size right, DNA all replicated okay, through mitosis, new cell division. Once the cells are divided, what are the possibilities? Well, once a new cell's formed, possibilities are they can continue to grow and divide. And you can think of populations within the body where that happens, the stem cell populations where that might happen. Can anyone think of a population where cells do continue to divide through adult life? Skin. Unless you happen to be my husband, in which case something's gone wrong and you ain't getting any more growth going on up here. What about blood supply? The hemopoietic system, continual division. The easy way to think of it actually is just to think of cancer. What are the symptoms of chemotherapy? Because what we're doing is blocking cell division. So people lose their hair because we're blocking cell division. They get nausea and diarrhea because we have stem cell populations feeding the gut. So you block those, so all the dead cells slough off, but they're not being replaced because they're on chemotherapeutic drugs. And they become very prone to infection because we're blocking in the bone marrow the reproduction of those cells. So in the adult, they're the populations that are readily continuing to grow and divide appropriately. They're the ones which we try and target with chemotherapy. We don't try not to target with chemotherapy, which normally are hit by chemotherapeutic drugs, however. The other thing cells can do is they can differentiate into a special cell type and cease dividing. So you can think of populations within the adult that do that. Many of the epithelial cells in the organs do this. What about neurons? Not going to divide anymore. So they differentiate into that neuronal structure and they stop dividing. And that, of course, is the problem that we have when we have neuronal injury, is that we can't, there isn't a stem cell population we can stimulate to repopulate that. They're the cells we say have gone into the G0 phase. They've differentiated, they're not going to come back into the cell cycle again. So they can enter senescence. That is, senescence is a lovely word, isn't it? Senescence is they do continue to exist, they're still active, but they'll never replicate again. And that's the fate of many of the cells. Once they're differentiated, they actually carry out their function and then they'll die. They themselves don't replicate to reproduce that population. And we say they go into senescence. The other fate that they have, as do all living things, is they can die. And there's two ways they can die. They can die via necrosis, which is a response to acute cell injury. And that's where the cell has undergone some very damaging event. Thanks, I know it's really frustrating for you, but I can really hear when you talk, and it's like, I almost start wanting to listen in. So, necrotic cell death is acute cell death injury. It's not pleasant. The cell basically just bursts and explodes and it dies. The alternative is apoptosis, which have I had the lecture for you guys on this so far, on apoptosis? I think so. Say yes and make me happy. Okay, so I won't take you through it anymore. So apoptosis is that form of very neat cell death. It's programmed cell death. It's genetically programmed. And the cell, instead of bursting open, releasing all its contents and potentially harming the surrounding cells, actually starts to break off its membranes and bleb and dies in a very neat way that doesn't affect its neighbouring cells. So this is the preferred method, but these are the possible fates for cells: continuing to grow and divide, differentiating, senescence, which is living, functioning but never going to replicate again, or obviously cell death. So that was a whirlwind tour through the process of cell growth and the normal life of a cell. Very, very condensed. Now what I want to talk to you about is what happens when things go wrong. And when things go wrong, nearly always what it ends up being is cancer. So let's think about cancer. What is cancer? Cancer is actually, as we came to study it, it's one that preoccupies those of us who are fortunate enough, we might say, to live in first world countries. It's a disease of a healthy society. So we know that it's something that tends to only occur later in life, predominantly. So you have to live long enough to get cancer. So if you're living in Bangladesh and you're starving, cancer is not something that you're really very preoccupied with, okay? It's not going to be a problem you're going to confront. If you're living in areas where infectious diseases are still endemic, it's more likely you're going to die of malaria than it is that you're going to live long enough to actually worry about cancer. However, in first world countries, I can pretty much predict for all of you at the moment, well, certainly for my generation, we will either die of cancer or some sort of cardiac problem, most likely, if not roadkill, but no, predominantly. So, and I've already picked which one I want. I'm eating my way towards a nice heart attack, is what I've decided. I've looked at the odds. Cancer, that, know a lot about cancer. Take that one, thank you very much. So, it's kind of fun to think about, why do I say this? And it's because when you contemplate the fact that I say it's a disease of a healthy society, you have to live long enough to get it, it actually has implications for why. And the why is because we now understand it's a multi-step process. You actually have to have many changes induced in a cell for it to become a cancer cell. And that takes time. So the reason I say it's a disease of a healthy society is that the epidemiology shows us that indeed that is the case. But what's the underlying reason for that? And that's because it is this multi-step process which we now understand. And it is usually detected many, many years after the first initiating events occurred. So looking at that, this is a diagram that I took from a Scientific American article. And I really liked it because it kind of took us through those steps leading to cancer. So what we have here is a nice layer of epithelial cells going very nicely on their extracellular matrix. They have a nice healthy blood supply here feeding them, and there you can see they have an orderly structure and shape. They're growing in a very orderly pattern. And what we now understand is among those, one of them will undergo a single genetic change. And what it does is it causes it to start replicating when it shouldn't. Still looks normal, still has the right shape, but now it's replicating more than we should. And that's the condition known, or you may know as hyperplasia. Cell still looks normal, but it's replicating more than it should. So we've assumed one genetic change. Something's taken the brakes off that regulation of how often the cell will divide. It's dividing more than it should, still looks normal. Among that dividing population, we think one of them undergoes another genetic change. And that genetic change further loosens its control on growth. It starts to grow more and more than it should, excessively. But now it starts to look not normal in shape, abnormal in shape. So here we have the ones that still look normal in shape, but these pink ones now look abnormal in shape, and they're also replicating more than they should. So dysplasia. Hyper for too much, hyperplasia, excess growth. Dysplasia, now they no longer look normal in shape. Now they're starting to be easier to detect. And among those, with time, one of them may undergo, so it's undergone this many, one of them may undergo another genetic event, rare genetic event. Now it has to be within that population, don't forget. Having the third mutation occur, that same identical genetic mutation occur in one of these cells will not necessarily lead to it being a cancer cell. But if it occurs among one of these cells, what do we see? Then they become even more abnormal in growth and appearance. They may now be growing far more rapidly and aggressively. They also probably now look quite abnormal in shape and appearance. Quite abnormal, very easy to recognize. Now if we're still confined to the site where this arose, that's what we will define as in situ cancer. It's still localized to the site where it arose. It hasn't spread beyond it. And it may stay contained in that state for years. Okay? Still just aggressively growing there, but not breaking beyond those boundaries. You can see this is going to be very amenable to surgical resection. It's still contained within here, you still have clean margins. So for a surgeon, if he can excise cleanly here, you can actually remove the bulk of the cancer, and that's what surgeons and pathologists will always look for. Do you have clean margins? That is, you've taken out all of it. And that, for that patient, the prognosis is going to be very good. You've managed to contain it and remove it all. However, among that population, one of them may acquire a further genetic change, and that genetic change is the one that allows it to start metastasizing. It's producing the proteins it needs, often they're metalloproteases that allow it to actually break through the extracellular matrix. And once it breaks through the extracellular matrix, it can then either invade the lymphatic system or the blood supply and start moving through the body. So this is a further genetic change it needs to acquire to do this. And the other thing you should notice now, we have a large tumor mass here. What else can you notice is going on? Angiogenesis. Now we have a large tumor mass, it needs to acquire its own blood supply if it is to survive. So really you have an evolutionary process going on. It now needs to evolve sufficiently to actually produce the growth factors that are necessary to stimulate angiogenesis. If it doesn't, the centre of the tumour will probably become necrotic and will start to die. And sometimes that's one of the problems when we start treating tumours, is that we have drugs that target rapidly dividing cells, which are the ones on the periphery that are getting the nutrient supply they need, the oxygen they need, removing the waste they need, but in the centre of the tumour mass they're not, so they're actually stop dividing. And those chemotherapeutic drugs and the radiotherapy don't actually affect those cells in the same way, because the centre of the tumour mass is actually quite distinct. The environment in there is quite distinct from the environment for these cells out around here. So you kill these, but these survive, and that can be one of the real issues for survival post-treatment. So what you now have is a metastatic tumour and one that has recruited its own blood supply. There was great promise with the anti-angiogenic drugs actually, and actually targeting this process, because in the adult human you don't have angiogenesis, you're not laying down new blood supply, and it showed great promise, but unfortunately with time, once you've knocked this off, the tumour does then tend to regrow. So it's something that needs to be in conjunction with other tumours. So what we've seen is there need to be several successive genetic changes in a particular cell for it to then give rise to this tumour mass, to give rise to the fully malignant invasive tumour. And so the thinking that I showed you is that you kind of have a clonal evolution of cancer. So you start with a normal cell, and as it divides, one of its progeny acquires the first mutation. The other one doesn't, continues to divide quite normally and is fine. But if this one, as it divides, if one of its progeny acquires a second mutation, and it has to be in one of these progeny, not in one of these, it then can start dividing even more abnormally. These ones will continue on, but it's these we're now worried about, because among those progeny, one of them may acquire a further mutation, and as it divides, one of its progeny might require a further mutation. So what you should think about, of course, is this is a lot of mutations. They have to be the appropriate ones that give the cell the properties it needs to start growing as a tumour. So, and they have to be the ones you need. So if you actually do the math, and if you say, okay, the likelihood of acquiring a mutation is 1 in a million, times 1 in a million, that's times 1 in a million, times 1 in a million, times 1 in a million. And the scientists did actually do the maths, and they worked out that actually, if that's the kind of rate that you need to acquire sufficient mutations to lead to cancer, it can't happen in the lifetime of a cell. Okay? It's too rare to get the exact mutation you need in this one, followed by in that one, followed by that one. And so they're like, it's really good, we've done the maths, you won't get cancer. We've worked out the odds. The likelihood, they worked it out, it's probably something like 1 in a trillion that one cell alone will acquire the exact mutation you need, followed by the exact mutation you need. So that's when scientists went, well, that's great, but actually people do get cancer. So we now think that perhaps some of the first mutations might be ones that actually lead to genetic instability in the cells, so that they acquire mutations more rapidly. And that's what helps them acquire all the changes that they need. Now, another way that cells can acquire all the changes that they need is actually exemplified by this. Have you seen karyotypes, cell karyotypes before? You probably have. So here we have a normal cell, and we've got false color images here using fluorescent patterning so that you can easily identify the cells. And here we actually have a breast cancer cell. And what you can see is they have wildly abnormal karyotypes, and they're frequently characterized by having far too many chromosomes. So you can see here, this is probably almost a tetraploid, tetraploidy. And not only that, so one way to get more and more of the activities you need is just to get more and more of the chromosomes that encode the genes with that activity. You'll also see there's quite a lot of translocations as well. This can also be characteristic of tumour cells. So that's one of the ways that the cells can do it. So when people started to look at, well, what are the kinds of mutations that you might need to acquire? This diagram was brought out and it said, here are the 6 hallmarks of a cancer cell. We actually think there's probably 8 now, but I'm just going to focus on these 6. So we talked about the fact that they must somehow be able to continue to grow without having a strict requirement for those external growth signals that we talked about. So they need to be self-sufficient in growth signals. And there's 2 ways they can do that. One is just not require them anymore, and I'll show you how they might do that. The other is to just make it yourself, sort of auto-stimulate. Need lots of growth factors? Well, every cell has a complete genetic complement that has the gene sequence for the growth factors that it needs. Why not just make it yourself and just produce it and thereby stimulate the whole population? We have anti-growth signals. Not only do we have in cells the ability to say go, the accelerator to turn them on, we also have brakes, checks and balances. So we also have anti-growth signals. And they just ignore those, they become insensitive to those. They evade apoptosis, so when the signal says growing inappropriately, undergo apoptosis, they have mechanisms by which they can avoid that. Limitless replicative potential, they actually reactivate telomerase, so they actually re-lengthen the ends of their chromosomes to make sure they can keep replicating them over and over without losing sequence. So they reactivate telomerase to give them limitless replicative potential. They upregulate the genes that are needed to produce the proteins so that they can actually break out of the extracellular matrix, as I said, survive in the blood and lymph, and then re-enter a different organ site to re-establish themselves at a distant location. And they activate, as you know, the proteins that are needed so that they can produce a blood supply as well. So if you think about it, these are a lot of genetic changes that a cancer cell needs to acquire to evolve into a very successful cell. If you think about it, they really have evolved into, they get to be the selfish one that grows and grows and grows and grows. And that's all the changes that they need to acquire. How do they do it? What causes those changes? Well, external factors such as chemicals, smoking, radiation, infectious organisms. We all know from epidemiological studies these contribute to the cancer burden. Individuals that are exposed to these or expose themselves to them are more likely to get cancer. The classic one that you'll all be so familiar with is smoking. We know that there are chemicals contained in the cigarette smoke that must induce those changes because those individuals end up with lung cancer. We also know there are internal factors. Some individuals inherit mutations that make them cancer prone. Excess exposure to hormones has been related to cancer. So we think part of the rise in breast cancer in Western populations is we have now a healthier diet. Women come into menarche earlier, go into menopause later. They have a longer lifetime exposure to estrogen. We think this partly contributes now, certainly to the hormonally responsive breast cancers. Immune conditions, if you look at individuals with AIDS, I don't know whether you know, but they're more cancer prone and get some rare cancers because our immune system does help protect against cancer. And oxidative metabolism. All the mutations that are induced by the fact that we live in have an oxidative metabolism. And here's the final take-home message. Ultimately, all cancers arise because of changes to essential genes that control cell growth and division. All cancer arises because of changes eventually to essential genes that control cell growth and division. And there they are, the ones that we just talked about. These are the ones that usually get changed. Signaling molecules, the receptors, these intracellular transducers, the transcription factors being upregulated, the cell cycle control proteins, lack of DNA repair, or the apoptotic proteins. So I'm going to come back and I'm going to give you a couple of minutes break. I'm going to take a couple of minutes break. Echo 360, the recording system will shut off. We'll start again exactly at 5 past, okay? Oh. Thanks, guys. So it's 5 past. Hello. Yeah, of course you can. That might help you if you like. I don't remember.